Okay, so welcome to the cardiovascular system. We did the blood or liquid portion of cardiovascular system. Now we got to look at the pump, which is the heart. And so this consists of the heart and the blood vessels that the blood is circulating in. And so the heart is this hollow muscular pump. It's basically going to force this blood around. And it will transport it through the blood vessels throughout the body. We have three different types of blood vessels. We have arteries, veins, and capillaries. Arteries, away, always away, regardless of whether they're carrying oxygenated blood or deoxygenated blood. Veins to the heart. And the capillaries are between the arteries and the veins. And we'll talk about, we have little arteries, arterioles, and little veins, venules. Okay. All right. So when we talk about the cardiovascular system, it is a closed circuit. And we have two circuits. We have a pulmonary circuit and a systemic circuit. The pulmonary circuit is to carry the blood that is oxygen poor because it's been used, the oxygen has been used by the tissues from the heart to the lungs, get that carbon dioxide dropped off, pick up oxygen, and bring this oxygenated blood back to the heart. And then once we do that, we want to get this oxygen-rich blood out to the system. And so we're going to do this in the systemic circuit. So we're going to transport the oxygen-rich blood out to the cells. We're going to release it to the oxygen to the cells. We're going to pick up carbon dioxide. We're going to pick up waste, and then we're going to make it back to the heart. Okay? So the way we work this is we have the right side and the left side of the heart. The right side is the pulmonary circuit, if you will, and the left side is going to the systemic circuit. So there we're looking at that. I remember we're in our chest. So this is the right side. This is the left side. Okay. All right. So you can see that oxygen rich and oxygen poor. All right. So in terms of the heart itself, about the size of our fist. All right. It is in the thoracic cavity. It is in the mediastinum, below the, behind the sternum, above the diaphragm, between the lungs. Basically, it's hollow, and it is divided into left and right halves. You can look at this anterior portion, and this is pretty good. This is the ventricles. This is the division between the right and left, and this is actually bigger, but it goes back behind the heart, so you can't see it. These are the atrium or auricles of the heart. These are just holding chambers, so they're not as big. This is the big pumping of the heart, is the ventricles. The auricles are holding chambers. All right, and you can see that location. And that's why if you do CPR and you're compressing here, you're doing a good job because that's where your heart is. It really is truly underneath there. All right, so we have a few coverings of the heart, and when we and these coverings, the terms for them are pretty universal, okay, in terms of parietal and visceral and that sort of thing. So I'm going to kind of go through this. So you have coverings over the heart. You have the tough outer layer that surrounds this double layer serous membrane, and that is the fibrous pericardium. And then you have two layers of serous membrane. And when you have two layers of serous membrane, like you have around the lungs, and we're going to be talking about this in the respiratory system, you have a parietal layer and a visceral layer. The visceral layer is always attached to the surface of the actual organ. The parietal layer is basically above that. Okay, so for the parietal pericardium, it is underneath this fibrous pericardium. It is what we call the outer layer of the serous membrane. Then you have a space. This is the pericardial cavity. And then underneath that, you have attached to the surface of the organ the visceral pericardium. When we get to the lungs, we'll be talking about the parietal pleura, the pleural cavity, the visceral pleura. Okay, so those terms remain the same. So if you get them down now, this will help you a little bit further on in the course. But you can see this, you kind of look, if you put your fist into a glove, this is what you will have, and you can see this covering. Okay, so there's the fibrous pericardium, the parietal pericardium, that space in there that is going to have the pericardial fluid, and then you have the heart. And the reason for this is because these are movable organs, like the lungs, they're going expanding in and out, the heart is contracting, and this helps us dissipate heat. It knocks down the amount of friction as these guys are moving. Okay? All right, the wall of the heart has 3 layers. The epicardium, this is the outer layer. It is also called the visceral pericardium. Remember, we said it's outside. So this is, again, to reduce this friction. Then you have the muscle layer, the myocardium. And this is fairly thick, okay? And this is made up of cardiac muscle. And then you have the endocardium, which is that inner layer. And it's thin. And it is so smooth because we don't want clots. We don't want anything to stick, the platelets to stick and form clots. So this forms the inner lining of all these chambers. And again, when I dissect the heart for you, you'll be able to see there is a true difference between the tissues between the myocardium and the endocardium. Okay, and you can see this on this graph. All right, chambers. We are mammals. We have a 4-chambered heart. Okay, all mammals have 4-chambered hearts. We have upper chambers for the atria, the lower chambers, which are the ventricles. We have little flap-like projections that project down from the atria. Those are called the auricles or ear flaps. Okay, think of it that way. So when you have these chambers, you have the right and the left. So you have 4 chambers. You have a right atrium, a right ventricle, a left atrium, and a left ventricle. Okay? So the right atrium receives the blood returning from the systemic circuit, from the inferior and superior vena cava and the coronary sinus, and pumps the blood to the right ventricle. The right ventricle then pumps the blood to the lungs. And then it gets oxygenated, comes back to the heart, to the left atrium, and then that goes down into the left ventricle. So, some of the structures associated with the heart, you do have septums. You have septums or divisions between the atria and between the ventricles. So there is a piece of tissue between the 2 atria that is called the intra-atrial septum. You have a piece of tissue between the ventricles, the interventricular septum. All right. You have 2 vessels coming in, 2 big veins coming in to the right atrium of the heart. That is the superior draining above the heart, the inferior draining below the heart, and that's going to return that oxygen-poor blood back to the heart. You have 4 valves. You have basically 2 different kinds of valves. We'll talk about this, but you have atrioventricular valves. These are valves that are between the atria and the ventricles. Okay? On the right side, it is the tricuspid valve. Try right. The way I kind of tell my students to remember this. Try right. And then on the left side of the heart, you have the mitral valve or the bicuspid valve. And it's named that because of the number of attachments. The tricuspid has 3, the bicuspid has 2. But tricuspid on the right, try right, mitral valve or bicuspid on the left. Then you have semilunar valves that at the exits to the heart, where you're exiting out of the heart going somewhere. These are semilunar valves. They are half moons, if you will, in shape. And you have two exits out of the heart. You have one going to the lungs and one going to the aorta. So the pulmonary semilunar valve is going to be as that, basically that opening into what we call the pulmonary trunk that goes to the lungs. And then we have the aortic semilunar valve, which is there going into the aorta. And sometimes it's called pulmonic valve and aortic valve. That's perfectly fine. We do also have what are called chordae tendineae, heartstrings. These are very fibrous strings that attach to the cusp of the atrioventricular valves, our tricuspid and mitral valve, and attach to papillary muscles that allow us to close those valves. We'll get into that a little bit later on. And then coming out of the pulmonary semilunar valve, you go into a pulmonary trunk and you go into the pulmonary arteries. But coming back from the lungs, you're coming back from the lungs via the pulmonary veins and coming into the left atrium. Okay, so you can see all this, but you can see the difference. These are atrioventricular valves. See the chordae tendineae, the heartstrings. This is the papillary muscles that contract. This is different than these half moons, which are the semilunar valves. There's the other one. There's the aortic, and there's the pulmonary. All right. So you can see that tricuspid valve. You can see that those three attachments. You can see the papillary muscles. You can see the chordae tendineae. You can see the cusp of the valve. Okay. All right. So one thing you do have to remember is you have to be able to trace a drop of blood through the heart. You've got to know this track. All right. So let's kind of go through this. Oxygen poor blood is coming in from the superior and inferior vena cava and the coronary sinus. The coronary sinus is the drain for the coronary vessels for the heart. Okay. And that all dumps into the right atrium. From the right atrium, it goes through the tricuspid valve into the right ventricle. That basically, we'll talk about this, the right atrium contracts, it sends that blood into the right ventricle, the ventricles contract, the tricuspid valve and also the mitral valve close, and the pressure rises in those ventricles, and it opens the semilunar valves, okay? When that opens, when the pulmonary semilunar valve opens, the blood flows into the pulmonary trunk, divides off and goes to the pulmonary arteries, goes to the lungs, drops off the carbon dioxide, picks up oxygen, and then comes back via the pulmonary veins to the left atrium of the heart. Excuse me, goes from the left atrium through the mitral valve into the left ventricle. And again, the right atrium and the right atrium and left atrium contract together. So when that contracts, that blood gets sent down into the left ventricle. The ventricles contract. That basically closes everything. The pressure rises. The aortic semilunar valve opens, and blood flows into the aorta. And that is your oxygen-rich blood going to the system. You need to know that. Okay? Now, the first branch off the aorta has to feed the heart. So those are the coronary artery. That is the blood supply to the heart to make sure that it keeps oxygenated. Basically, the branches, you can see them. There's the posterior interventricular artery, the marginal branch, the circumflex off the left, and the interventricular, the anterior. Okay, so that's going to supply it. Then you're going to have cardiac veins that are going to drain that blood from the myocardium, and it's going to go into the coronary sinus, which runs around the area of the heart between the atria and the ventricles. Okay, and you can see this right here. This is your coronary sinus right in there. Okay, and you can see that piece. Okay, so let's look at some heart actions. First of all, some terms: systole, diastole. Systole means the heart chamber is contracting. Diastole means the heart chamber is relaxing. So we're coordinating this. So you have what's called a cardiac cycle. These are the events that occur during a heartbeat. You have the atria contract while the ventricles are relaxing. So you have atrial systole that occurs while you have ventricular diastole. And then the ventricles contract while the atria are relaxing. So you have ventricular systole during atrial diastole. And then you have a very brief relaxation between the two. So let's talk about the muscle cells that are doing this. You learned in AP1 the difference of cardiac muscle from skeletal and smooth muscle. So you have one central nucleus. It is branching. You have the intercalated disc between the cells, and they contain these desmosomes so that you have support. You have a gap junction so that this information spreads very quickly. Okay, and basically you form a functional syncytium, a mass of cells that functions as a unit. All right, so you have the atrial syncytium and the ventricular syncytium. You want them to contract as one. All right, so you can see these gap junctions and those intercalated discs. So the conduction system for the heart, again, you have these specialized cardiac muscle cells that are going to initiate this potential. You have the sinoatrial node, or the SA node. This is the pacemaker of the heart. It is genetically encoded to initiate an action potential at a regular rate. This initiates the rhythmic contractions of that heart, okay? And you have and then it basically moves from the SA node, the sinoatrial node, down to the AV or atrioventricular node. And then that will be conducted to the atrioventricular bundle of His. And basically, then it will go down the bundle branches and the Purkinje fibers. But bottom line, you're basically sequencing this through the heart. So as it moves from the SA node to the AV node, you get a slight delay right there at the AV node so that the atrium can contract and drain the rest of that blood that's moved into the atrium so that we get most of that blood down to the ventricle. The whole idea is to get as much blood out of the heart as we can. So we squeeze the atria to make sure that that occurs. And then we go from the AV node down the bundle. And that's basically what we want to do is we want to very quickly, boom, swing it around the heart so that the ventricles, which are fairly large, contract together. So it's going to very rapidly conduct these impulses. And then we split into bundle branches so that we hit both sides of the heart. And then the Purkinje fibers are these large fibers that conduct these impulses to the myocardium and then up again. Okay, so you can see this from the SA node down to the AV node to the AV bundle, and then we split into the bundle branches and then up the fibers so that we get everything contracting as one. Now, how do we know this is occurring? Well, we have an electrocardiogram, an EKG or ECG, depending upon how old you are. The older people will say EKG, the younger people are being taught ECG. It doesn't make any difference. But what we're doing is a recording of the electrical changes in the heart. Okay, and what we're assessing is the ability of that heart to conduct impulses. If you actually read an EKG, what you're really looking at is the timing on this, because the chart is a timed chart. And so we're looking for how long does it take to go from point A to point B to point C to point D. All right. Well, there's several waves in here if you look at it, but if you notice, you see these milliseconds. This is the timing. So each one of these has a timing, and that tells us, is it the correct time? If the time is wrong, then something's wrong in the conduction through that heart. Okay? So the first wave is the P wave, and that represents the atrium depolarizing. And we all know this because we took this in AP1. If you've forgotten what depolarization and repolarization are, you need to go back and review this, okay? But the atria, depolarize, okay? And again, this occurs right before the atria contract. Then you will see three waves, the QRS complex, and this represents ventricular depolarization. There is at this time, atria are repolarizing. But because the QRS complex is so big, we can't see the repolarization of the atria in there. And then we run to the T wave, which is ventricular repolarization. Okay? So you can see there is, and you can see it as it goes, and you can see what's happening. But there's your P wave, there's your QRS complex, and there's your T wave. And you can see hidden in here, again, would be the repolarization of the atria, because this is just so huge. Okay? But that's what we're looking at this conduction, and we're looking to see how long this occurs. Another evaluation of the heart is to just listen to the heart itself. All right, we're listening to the heart valves close. We're listening to vibration. We're listening to the blood flow. So some of the heart sounds that occur, everybody thinks that ba-da, da-da, da-da. They say lub-dub. I don't know about that. But the first sound, which we consider S1, sound 1, occurs during ventricular systole. The AV valves are closing. The second valve, S2, is associated with the ventricular diastole, and the pulmonary and the aortic semilinear valves are closing. And when you hear what's called a heart murmur, this is because you're getting an abnormal heart sound because the cusps of that valve have not closed properly. Okay, so you can see what's going on here. And you can look at, this is a nice little chart that looks at what's going on with the pressure as the pressure rises, what valves are opening and closing. And you can see the pressure in the heart and what's going on in the heart in terms of the pressure in the aorta, the ventricular pressure, and with these valves opening and closing, and what the EKG looks like and what you're hearing in heart sounds. So spend some time with this. Think about this. Ventricular diastole, what's going on? Okay? There's the P wave. That's the initiation. So we work all the way through so that you understand that. All right, so during the cardiac cycle, the pressure rises and falls. When you are basically early in ventricular diastole, the ventricular pressure is lower than atrial pressure. The atria and the ventricles are relaxed. The AV valve is open. The semilunar valves are closed. The AV valve is open, and the blood is just flowing from the atria into the ventricles. Okay? And then you have atrial systole. And that's when we squeeze those atria and we squeeze out the last part of that blood and send it into the ventricles. So the ventricles are relaxed, the atria are contracting. The AV valves are open, the semilunar valves are closed, and we'll talk about how they close. Atrial systole just basically pushes the remaining blood that's in those atria. There's a little bit left, but it into those ventricles, and that causes that ventricular pressure to increase. When the ventricular pressure rises above the atrial pressure, the AV valves close. The chordae tendineae prevent the cusp of the valve from bulging too far back up. That's the whole idea. The atria relax because it's done its job. It's pushed the blood down into the ventricles, all right? And blood flows into the atria from the vena cavas, the inferior and superior vena cavas, and the pulmonary veins, all right? And the coronary vessel. All that stuff goes into those atria to get the blood coming in. The ventricular pressure will continue to rise. You get an isometric pressure rise, and then you finally overcome it, and the pressure increases enough that it pushes open the semilunar valves. And the blood flows out of the heart into the pulmonary trunk and into the aorta. Okay? And then when those ventricles relax, those semilunar valves are closed because the blood will back up. And again, those are like little half moons. They're kind of like umbrellas. And as the blood flows into that valve, it opens up and it prevents blood from backflowing into the heart. Okay? All right. So what regulates this? Well, the body's going to regulate the rate and the volume depending on what you're doing. There is a cardiac center in the medulla that looks at this regulation. Normally, the SA node will control the heart rate. It is genetic. It has a genetic rhythm to it. And the heart rate changes when there are certain factors that influence that SA node. All right, it can be physical exercise, it can be body temperature, it can be fight or flight, epinephrine, concentrations of potassium, calcium, those sort of things. So that a lot of things come into play there. Now, when you sit there and look at it, think about it. Sympathetic, increases heart rate. Parasympathetic, rest and digest, lowers it. So parasympathetic impulses reach the heart via the vagus nerve. If we remember, what cranial nerve is that? And that lowers that SA node rate to about 60 to 80 beats a minute. And it's just because it rests and digests, you're going down lower. If you have a sympathetic impulse, that's going to raise it. And that's going to raise that heart rate. You have epinephrine flowing, it's going to raise it. You do also have these reflexes, a couple reflexes. You have baroreceptor reflexes. These are in the medulla oblongata also. And basically what you have is a cardio-inhibitory reflex center and a cardio-accelerator reflex center. And basically, this is baroreceptors or pressure receptors. We are looking at is the pressure too high, the pressure too low. Where are we looking? We're looking in the aortic arch and the carotids. They're detecting the blood pressure in the carotid arteries and in your aortic arch. All right? So if this pressure gets too high, if you're getting this pressure sensors are being hit upon, it will lower the heart rate and lower the blood pressure. You have stretch receptors in your vena cava, and the same thing works. All right, you may need to lower venous pressure. So you have several sensors that are working. The other thing that affects it, of course, body temperature is going to raise it. The levels of potassium and calcium again may change it. but you've got a lot of different pressure sensors looking at what do you need, and do we need to raise blood pressure, do we need to lower blood pressure. All right, some interesting things that can happen. You can have different arrhythmias, different heart rhythms from normal. I'm sure you've heard some of these. Fibrillation, atrial fibrillation, all right? This is an uncoordinated contraction of a small area. Atrial fibrillation, this is just kind of vibrating, like this. It's basically beating, but it's not life-threatening. It's chaotic contractions. And it's not life-threatening, but ventricular fibrillation can be fatal because atrial fibrillation, not so bad. AFib, you can get clots and stuff like that, so you don't want it. But ventricular fibrillation, if you don't get a coordinated contraction of the blood to the rest of the body, that's a problem. Tachycardia, heart rate over 100 beats per minute at rest. Bradycardia, below 60. If you're an elite athlete, it can be 45, and that's perfectly normal for you. All right. You can have flutter, which is rapid contraction of a heart chamber, 250, 350 beats per minute, atrial flutter. You can get PVCs, these premature preventricular contractions, PACs. This is where the beat occurs before you expect it to occur. And it originates from what we call ectopic regions of the heart. This is sometimes where you have extra cells within the heart that are actually sending out signals that are interfering with the signal coming out from the SA node. So you may have additional beats, okay? And a lot of times what we have to do is an ablation, kill those cells, or we may have to put in an artificial pacemaker to make sure that the rhythm is the correct rhythm. Okay, so let's look at our blood vessels. Blood vessels, they are closed circuit. You have several different types of vessels. You have arteries, arterioles, which are little arteries, and then you go into capillaries, in this capillary bed, if you will. And then you have on the other side of the capillaries the venules, which are little veins, into the bigger veins. All right, and that's the way it gets transported. So you can take a look at these and you can see some of the structure of these. They are different in terms of what they need. So if we're looking at arteries, they're thicker, they're stronger. They have usually 3 layers in terms of that. Arteries have to take the push of the blood coming out of the heart. So they've got to be fairly elastic. So you have these layers. You have an innermost layer, which is the endothelium, which is nice and smooth. And then you have a smooth muscle layer with elastic tissue in it, the tunica media. And then you have the tunica externa, which is the outer layer with connective tissue. And so we're transporting this blood under high pressure. And then as we go further away from the heart, we give rise to these smaller arterioles, which are thinner and smaller. The walls get thinner as the arterioles get smaller, but they can still undergo vasoconstriction and vasodilation. You can see this smooth muscle. You can see this precapillary sphincter. So if we want to go to the capillary, we can. If we don't need to, we don't have to. The capillaries are the smallest diameter vessels. They do connect the arterioles to the venules, and they are extensions of this inner lining of the arterioles, and they are just simple squamous epithelium because this is where exchange has to occur. So you have to have a thin membrane. These are semi-permeable. You exchange the substances between the blood and the tissues, and this is regulated again by these sphincters, and you have the smooth muscle that surrounds that so that you can do whatever you need to do with it. You can see this meta-arterial, and you can see the capillaries here, and you can close these sphincters and go through these capillary beds if you don't need to or not. Or you can completely just shunt and bypass this particular area. So when we look at capillaries, we have openings in the walls of capillaries. They're slits. And we have different sizes of openings. So we have different types of capillaries depending upon what's going on. We have continuous capillaries. They have very small openings. This is found in muscles and connective tissue and skin. We have fenestrated capillaries. These have pores. These have large openings so that you can move a lot of stuff across this. You see this in the small intestine, particularly in the kidneys and in the endocrine glands, so you can get large amount of things across. And then you have the sinusoidal capillaries. These have the largest openings. These show the spaces between cells and cavities. These are found in the liver and the spleen and the bone marrow, where you're actually looking at the blood and the tissue coming through there. In the spleen, when we get to the immunity, this is where we're looking to see if we have any invaders. All right. So you can see these fenestrations, and this determines what stays on one side of the membrane and what goes through the other. It's kind of like a colander, if you will. All right, so the capillaries, the higher the metabolic rate of a tissue, the more work it's doing, the denser the capillary network. So you have a rich supply in muscle and nervous tissue, since they're using a lot of nutrients, they're using a lot of oxygen. Areas like cartilage, they're not going to have many. And the blood flow distribution to the capillary can change again with the situation. If you are exercising a particular area, these capillaries receive more blood flow. All right. And the digestive system receives less. It depends on what's going on. After a meal, digestive system needs to receive more. And so you will change it around depending upon what is happening. And again, we had that precapillary sphincter, that band of smooth muscle that wraps around that to regulate blood flow. If you are injured in a certain area or you're cutting off flow to a different area, you can bypass that area.